Subio Platform can manage many multi-omics datasets. You can separately store mouse data from human or other species with the organism level. In the mouse organism, there are several microarray platforms and general gene ID platforms for RNA-seq datasets. The series associated with the selected platform are listed. You can filter series by keywords that specify the study. If you find a series that you want to analyze, load it. The Analysis Browser tab opens automatically. The Sample Info tab provides information about the study. You formally filtered the series based on this information. You can click on the link to open the website and double-click the attached files to open them. Without the Subio platform, you often see the tables of differentially expressed genes with Excel. By the way, you can modify this annotation table by showing, hiding columns or changing the order of columns. You can, of course, sort the table by fold change or p-value, like Excel. Or you can search by keywords. But that's almost all you can do if you use Excel. You can view heat map images on a PDF report. Or you can view several pathway images on a PDF report. But that's all. You can't dig deeper into the dataset. Is it enough for understanding or interpreting? Subio Platform offers a higher level of experience. You can check the raw data quality and every step of preprocessing. The uniformity of the distribution patterns indicates that the experiments went successfully. You can see the boundaries of the noise and signal ranges in the scatter plot, and the low signal cutoff is appropriately set. The ratio to control samples block converts normalized signals into log2 ratios relative to the control group average. There are lists of differentially expressed genes. You can not only view the table, but also do more with the Subio platform. For example, you can separate genes that show the common expression patterns by their expression levels. Some genes are highly expressed, and others are moderately expressed. Interestingly, some genes are almost not expressed when pional is added. Comparing gene counts may tell you more than fold differences because they might behave like on/off. You can save the genes as a new list. And you can do it like this as long as you get ideas of genes' behaviors. Moreover, you can search by keywords among the collected genes to find genes of interest. You can work with the results of clustering analysis, not just view them in a PDF page. If you find a cluster of an interesting expression pattern, you can select it by clicking the tree node and save it as a new list. Of course, You can separate these genes by expression level and extract on/off behavior candidates, as in the previous example. Another exploration is to examine the chromosomal location bias of the selected pattern. You can drag a range along a virtual chromosome to view the expression pattern there. You can view not only genes of the selected expression patterns, but also other genes in the area. Now you find a cluster of genes that behave similarly. You can drag the area to see what they are. In the pathway view, you can drag around elements to see what they are. Alternatively, you can highlight pathway elements using keyword searches. Or drag to select receptors on the cell surface to see what they are. Pathways often connect to neighboring pathways, allowing you to go further. You can jump from one pathway to another as long as you are interested. Sometimes, an element consists of multiple genes. You can see their expression patterns one by one. And you can check the expression pattern with the line graph, or search for the gene on the web by right-clicking. Of course, you can add keywords to target specific websites. Lastly, I'm showing how easy it is to hand the whole data to colleagues or collaborators. All you have to do is export an SSA file and give it to them. If they receive an SSA file, drag and drop it onto the Series tab in the lower panel of the Data Manager tab. They can explore the dataset on their computers as they like. All data you passed, including DEGs, clustering, pathways, and attachment files, are ready for further analysis.